So hi everyone, my name is Evangelos Karadzias. I'm a postdoc fellow in the Rob team, RobFin group. It's called MGnify, or otherwise it's known as Microbiome-Informatics. And today I'm going to present you our newly released MGnify Protein site, along with 2.5 billion protein resources that you can finally query and get your hands on some data and metadata from MGnify. So our goal for today is to gain some skills, and I've put them here in the beginning of the presentation so we know from the start what we aim to achieve, and we'll be covering them one by one. So the first thing is, after this webinar, I would like that everyone, I would hope that everybody is keen on finding the various MGnify protein related resources. And this will be the MGnify protein site itself, the flat files that we provide, an online database to query, as well as specific protein family resource called MGnifams. Another thing that I want you all to be able to do afterwards is to be able to, for a given MGnify protein, I want everybody to be able to explore the environment coverage of that protein, where it is found, to be able to find the domain annotations of that protein, as well as find and see the 3D structure of that sequence as well. Then the most important thing, I think, from this talk is the third point. So performing a sequence search against the MGnify protein cluster representatives. This will allow anybody that has a protein sequence of interest to be able to explore it further by trying to find hits against the MGnify proteins and be able to retrieve metadata, gain some knowledge on what the protein is about. And finally, hopefully learn how protein families can be generated out from a set of starting protein FASTA files. So the contents, very briefly, will be a very brief introduction to MGnify for those that are not familiar with it. Then we're going to see the newly released site called MGnify Proteins, and I will show you how to do sequence searches through it. Then we'll see some alternative options to access these MGnify proteins through flat files and a database. And finally, we will explore a specific use case on how to build upon this set of proteins, in this case, the MGnify's protein families resource. So MGnify, what is MGnify? MGnify is a platform for the assembly and annotation of microbiome sequences. One can go to the main site, this is the main user interface, and can submit their own data. They will have to go through the ENA portal first. ENA is the European Nucleotide Archive. So every data that is submitted is standardized before we process it in MGnify. Or if they don't have their own data, they can also request data that already exist in ENA to be analyzed through our pipelines. So why should somebody do that? Our analysis offers insights into the microbial diversity. This means the taxonomy that is found in the samples, as well as the potential functions of the samples and the proteins, and also see the different biomes samples are found in, and be able to search the database, the MGnify database afterwards through selected biomes. We offer various different analysis types, and we have a collection of standardized pipelines and tools to do that. Everything is publicly available through our GitHub page. I won't go into too much detail in all the MGnify pipelines for today, because we're aiming to do proteins mostly. And these proteins come through this 60,000 up to now assembly analysis that we've done so far in MGnify. So zooming in a bit, this specific pipeline. So how do we go from analysis to this resource of proteins? We have many different samples of various biomes. I will use the term biomes and environment interchangeably. So all these input samples are accompanied by data and metadata that the user have uploaded. We sequence all this data, the raw reads, And after we have the reads here, we use assembly tools to produce the various assemblies. And this is the point where the protein prediction itself is happening. And we specifically do that with a combination of two gene calling tools. These are prodigal and frag gene scan tools. So after analyzing the 60,000 assembly analysis, we end up with a protein sequence database of almost 2.5 billion protein sequences. These, along with their metadata, can be explored through various resources now, and we'll go into more detail for this one by one. So starting with the MGnify Proteins site, this was released recently, if I'm not mistaken, 2 months ago for the general public. And as I told you, we have 2.5 billion sequences in the back of our databases, but for the site, we have clustered these sequences using some thresholds of 90% coverage and sequence identity. So in the event, we present in this site, and for the user searches, the 700 million cluster representative sequences, both to view and search against in the site. The starting set of 2.5 billion sequences were already non-redundant, but we did this because still some of them might look alike, so we didn't want to waste resources of predicting several similar structures and having almost redundant information. So yes, this is the starting landing page of the MGnify protein site. Here in the example, we have already searched with a MGnify protein identifier. And the first thing we see down here is how big the cluster size is, because as I told you, remember, these are all cluster representative sequences. So we're talking about cluster level now. And then we can find the extra information, for example, if this protein comes from a full-length open reading frame, or if this was a partial protein. So X here means that this is not a full protein, just partial. Then again, next to it, we can see the biomes and how many times this protein was found in this biome. And then we also have some visualizations on domain annotations on the specific sequence. For example, this here has this protein family. And here you can see at which point of the sequence itself. So we can see this protein family annotation towards the end of the sequence. You can follow the site either through the link, or you can use your phone to go to the site, or you'll get the slides afterwards. But for now, I'll just click on it. So we'll have it open here next to the presentation. And I will go back to it in a bit. So let me finish showing you the user interface here. Other than the domain architecture, one can find the 3D structure of the protein, the predicted structure. This was predicted by using the ESM-fold software of the meta-AI team. And also one can trace back this particular sequence to the original study and assembly from MGnify. Of course, the 2D representation of the sequence is also available to the user. So let's go back. This has mostly covered the second part of the skills to acquire, but let me also show you interactively in the site. Go back. So if you've already searched MGnify Proteins, this is the starting page. You can either search by identifier up here or do your own search query down here. We'll see this in a bit. So I have already opened a more, let's say, intriguing protein here. Again, it's a small cluster, it's a singleton, only one protein, but this has more than one PFAMs annotated on it. So you can see here, it's interactive. One can zoom on the PFAM visualizer here and can move this into different parts of the amino acid sequence. And also, if you click on a specific amino acid, this will zoom in the protein structure itself down here. The other way is also the events are linked. So if somebody wants to click or scroll in the structure, they will also see which amino acids on the domain visualizer this corresponds to. So this is a pretty useful tool to see exactly where the domain starts, both on the sequence and on the structure itself. And of course, one can trace back the assemblies and studies in MGnify. This is also important. We can see information on where these samples come from geographically, as well as the metadata that the users uploaded when they uploaded their data. But there are not many cases that you already know MGnify protein identifier to come and search for it, right? So the most useful thing to do is, I'll go back to the presentation for now. So the second way to query the MGnify proteins is through inputting your own protein sequence, amino acid sequence. There are various user thresholds that the user can select here. And after the sequence has been pasted, you press the search button, and then you're transferred in the PIHmer site and can see the results. So you can see the various MGnify proteins with their identifiers in the results here. And all these are clickable and link back with the MGnify ID back to the MGnify protein site. And here, with a glance, you can find information about your input sequence query and can see some PIFA matches and the relative scores. So to see this example as well, we can go here, just use this example. We can give some fewer amino acids if we want. You don't have to have spaces here, it's just formatted in a way. It can have a smaller name as well. And then press the search button. So the user is transferred to the PIHmer site, and the results should be loaded here. I have already, do not waste time, I've already loaded the results here. No, not same results, sorry. Okay, we can leave this loading a bit. Yeah, there they are. So these are the MGnify proteins with their IDs. If you click here on the arrow, you can see the alignment as well, the query protein and the target protein from MGnify proteins, and also get links to the studies and assemblies, as well as the PIFA matches. So if we click on the MGnify protein, then we're transferred back to the original MGnify protein site, and we can find some extra information. Some of the proteins don't have predicted structures. We do that for storage purposes. And yes, you can keep exploring its biomes, cluster sizes, and also PIFAMs. If you click on the PIFAM link, this takes you directly to the Interpro website, and you can see more information about this specific protein family. So, going back in the presentation now. We've already explored skills number 2 and number 3. We've seen the MGnify protein site from this first goal. And now let's take a look into the two alternative resources that one can use to access the MGnify proteins. First is the FTP site. This is mostly for developers, I would say, because flat files are quite difficult for a simple user to parse. Information about the generated files from our pipelines can be found in the README, but in a nutshell, we have information about the assemblies, the biomes of the sequences, the clusters themselves, which sequences are in which clusters and how many members there are, contig mapping for the sequences, the proteins themselves, as well as additional metadata regarding contigs and PFAM annotations. Also, very recently, the database itself, the whole database with the 2.5 billion sequences was released in Google Cloud, BigQuery. This is freely hosted for us. And I have left a link here with how to create an account and use that freely available database. And this is not very easy. I've created I will do a demonstration now, and I will give you some hints on how to use it because it's freely hosted, but after your free trial expires, you will have to pay some credits in order to run some queries. So I will walk you through it. Let's do that now. First, before going to the Google Cloud database, I just wanted to show you, this is the link for the FTP site. You can access the README here, find information about all files included in this folder, and extra information about the total number of sequences here, how many of them are full length or partial, and so on. Now, let me go to the BigQuery one. This is the database. As I told you, it's freely hosted in Google Cloud. And if you press View Datasets, you get into this site. You just need Gmail for this and register for a project, free project up here. Now I created the project named as MGnify Proteins. There is extended information on how to do that in the how-to link that you will be able to find in the slide. But to keep it short, you select the EBI magnified database from the left side here, and you can see the tables of the database here. What we care about mostly is the protein table. And if you click on the protein table, you can see the various table columns. And in order to query this, you just press here in the query, you can select split tab or new tab, and then you have a bit knowledge of how to use queries or ask ChatGPT how to do it. It's not difficult. If I do this, this means select everything, all columns, from the protein table hosted online and limit it to 1,000 first entries. So what you need to take care of, this is very sneaky. Here it will tell you on the right side, it will tell you how many gigabytes will be processed if you run this query at the end. Don't try doing this yet because the indexing of the protein table was not correctly applied by the Google team, and we're trying to work on that. So you'll have to wait a bit more because your free monthly credits is 1 terabyte. So one query in this table will just deplete all your free credits. So don't do it. Let's go back to the presentation. So getting there, we already saw 3 of the 4 sites for today. And now I'm going to talk about some use cases that you can build up on top of MGnify Proteins. What has already been done is, first of all, ESM Atlas. If you don't know that, it's the software I showed you that predicts, that generates the output I showed you, the 3D structures. The Meta AI team trained their models on our MGnify Proteins, and created the ESM Atlas resource that stores all these MGnify protein structures. Another use case is plastizymes, searching for plastizymes. These are novel plastic degrading enzymes, and we have a bit of those in MGnify Proteins. Just some use cases to work your imagination. Then leveraging the genomic context information in our databases to be able to link gene locations with functions. And finally, last one is build metagenomics derived protein families out of these protein sequences. This is my personal project, so I'm gonna go a bit deeper into it. This is called MGnify's very imaginative name. So yes, why do we care about this? We already have protein families, right? But the more sequences we add, we can expand the current protein family sequence space, and this will help in aiding the characterization of these families. We already have them, but we might not be sure exactly what they're doing, so we can explore them a bit more. But other than the known families, we might be lucky enough to identify new novel protein families that have not been studied before. And what exactly are we doing in this extension, in this use case? We do protein family generation. So we go from microbial sequence clustering to family model generation, and also try to annotate all these families, either via profile matching against PFAMs, already known PFAMs, or predicting their structures and then using them to find distant homologs in structural databases. So a brief overview, we'll go very fast through the pipeline of MGnify. So how are we doing this analysis? First of all, we start from the initial dataset of 700 million cluster representative sequences that you can also, they are the same that you can also search against in the MGnify Proteins site. We slice off known PFAM domains, so we don't just reproduce the already existing PFAMs and keep those sequences that have more than 100 amino acids. Then this leaves us with 235 million input sequences for MGnify. Then we recluster them again in new clusters, but with a lower sequence identity threshold to go from just very strict clusters to, let's say, family level clusters. And then this leaves us with almost 200 million initial clusters, from which we will focus on those that are at least with 50 members, and this leaves us with 18,000 clusters. We take all this through the family generation algorithm. I will go into more detail in a bit. And we generate, in the end, 6,000 non-redundant MGnify. After we have generated the models, we do the annotation here with the blue. It's all about annotation. This is profile matching with the HH-Bliss software against PFAMs. Then we predict the structure using the ASM fault software, and then we do homolog detection with the fault-seek software against PDB, AlphaFoldDB, and DSM Atlas databases. So yes, let's focus a bit in the family generation algorithm. We started from 18,000 clusters, right? Then the first step we do is create the multiple sequence alignment with the math software. Then we build the HMM model with HMM build. Then we recruit more sequences from the initial dataset with HMM search, align this to the, if there were any new, to the multiple sequence alignment. And if there were no new sequences, we just exit, or we remove redundant ones and keep doing the same thing like up to 3 times. I won't go into more detail and bore you with this stuff. Sorry, it's my project, so I want to talk about it a bit. So we already have a site to present these results. It's quite similar to what I showed you for MGnify Proteins, and the two resources are linked with each other. So if you aim to use MGnify Proteins to build your own stuff, it would be very nice if you could link back to the original site, right? So for this example here, I have a random family that has 1,500 sequences inside. And then this is the link to the original sequence that it came from, as well as its slice here. Where does it start on that sequence and where does it end? So if you click this link, you will be taken back to the MGnify Protein site and see with yellow, both on the structure and on the 2D sequence, where this particular domain, let's say, falls upon. Then we offer some extra information about the underlying biomes of the total family sequences and the underlying domains as well. This means here that 390 sequences don't have any other domains other than this family itself. And then 299 sequences have the MGnify 74 following 2 glycosyl hydrolase protein families. Then on our predicted structure this time, we also annotate the predicted scores. The more blue the amino acid is, the better the score. And if it's orange, well, not so great. Also, we present the multiple sequence alignment visualization and the HMM itself. We'll do a demonstration on the site afterwards, so we can explore a bit more interactively. And if you hit this submit HMM search button, the user is then taken to the Hammer site, where you can see similar domains on PDB sequences that already have their PFAM annotations. Finally, we present 2 tables with both the profile matches and the structural hits. Everything is linked back to the original resource, and there are some more information like statistical values, the significance of this match, as well as where it starts and where it ends. Both on the protein families and in the target sequences, both query and target sequences. So I think I've talked about everything by now. So let's just do a brief demo on the MGnify side. This is another protein family. It's the first one. No names yet, just identifiers. This is also quite newly released, so we haven't been able to go through all 6,000 of them and give them some more meaningful names. So yes, as I told you, you can click on this link, go back to the original sequence. This is a bigger cluster with 72 proteins. All of these were found in the digestive system. This protein was found one time in the digestive system. This is what it means. And then here with yellow, we can see the specific MGnify, where it falls upon the whole structure, the whole sequence structure. So one can play here with the structure again. If you mouse over the structure, you can also see the event here on the 2D amino acid sequence. This has quite a good score of prediction in total. And then one can view the multiple sequence alignments here and the HMM models here. What does this mean? This means this explains what's the probability of each amino acid being in that particular spot on the sequence. So if you click here, you can see these probabilities, and it will also highlight where you are on the model. It will be highlighted in the multiple sequence alignment as well. Finally, the domain architecture here, I already explained that. This is also linked. If you click on this, you can be transferred to the specific protein family in Interpro, get more information about this PFAM. And finally, can click on the similar structure that was found as a homolog here and see how well it matches this protein family. A bit simpler, yes. So with that, I will finish for now. I would like to thank my team. And yes, feel free to ask any questions.
Great. Thank you very much, Vangelis, for the presentation today. And thanks everyone for joining the webinar today. So the first question is, when using fold-seq to search by structural similarity, is the MGnify ESM30 version 1 the same database as the MGnify Proteins mentioned in this seminar?
Yes, that's what's used. I don't think there is another version of ESM that you can use with fold-seq.
Okay, great. Thank you. Can one provide a profile HMM already made locally instead of single sequence to search the MGnify protein database?
I think that can be done, but not through this site. It can definitely be done through the API, but not through the MGnify protein site.
No, but you can use the sequence search. You just posted the answer there. You can use the sequence search and upload.
So we have another question. Is there information available how a specific biome is defined? For example, the alkaline biome, which pH can be expected.
Not directly on the protein side. We don't have metadata around the study in this resource, but you can get the information about the study, that sort of information from either ENA or even our API. So you will need to pull data from different places in order to get that.
Yeah, this is what originally the person that submitted the data originally wrote down. We just carry it with the metadata.
Okay, thank you. Is there any general comment you'd like to make, Evangelos or Martin?
I hope that people understood what I was talking about, and after this, we'll be able to use the resources.
Yeah.
Just feel free to poke us anytime, email us. We'll be able to help.
All right. So if there are no further questions, we can end the session today. So, all right. So thank you, Evangelos and Martin again, and thanks everyone for joining. Goodbye.
Thank you. Bye-bye.